Want to learn and get your money's worth. You're paying for me to entertain you. And entertainment is not really the same as learning, but if it's fun, it's more likely to be learned. So think of it as entertainment, not as lecture. Spring 24, not going to help you any. This is still this January 29th meeting. It's not going to help you either, but if you're not familiar with the Women in STEM program that started last semester, last year. It's for men and women, is to try to promote getting women into STEM things, so like biology, engineering, computer science, things like that, where it hasn't really seen the same types of gains as health related to those staff. Christina Pitts will probably come by later this semester and talk about it. You'll hear a little more about it. Does anybody know who Christina Pitts is? If you don't, she's the pre-health sciences pre-professional health sciences coordinator, which means she's trying to help you get into med school, dental school, and stuff like that. But she has other information. Hopefully she'll come by and talk to you about it also, just for fun. It's always a question, what do you do if you don't get into med school? But there are other options. There was a professor, really old professor here, that said, the only way you're getting into med school is as a cadaver. That's really rude. It's pretty rough. So we don't say that anymore. Anyways, that's about 2 minutes of wasted time. So, here we go. We left on simple and complex traits. Simple traits, remember, one gene affecting a single phenotype, which means trait, so one gene affecting a single trait. Complex traits are where we have multiple genes affecting the phenotypes that we see. Morphological is neither simple nor complex, it could be either one. So we do see examples of morphological traits that are simple and complex. Morphological and molecular are the ones that we will talk the most about this semester. We don't really spend a whole lot of time on talking about physiological traits. Definitely not behavioral traits, because that's not really the point of this class. Maybe in ecology and evolution, you might talk a little more about behavioral traits and how they seem to work. For morphological traits, I kind of think about them as being cosmetic. I don't want you to get the idea that cosmetic is bad or not useful, because some cosmetic traits are actually very beneficial. Having a certain plumage is very desirable to attract a mate. So it might not help in the survival sense. Some traits do help in the reproductive area. You might not think about hair color being important, but who goes for somebody with a certain color hair? Just like, yeah, I like blondes, I like brunettes, I like redheads. Nobody? Can't. Just up here talking. Okay. But morphology means shape, appearance, form, structure, whether you have one horn, two horns, in the front, on the side, on the back. Anybody know anybody with horns in the back? Stegosaurus. Boom. Although not called horns, they're called plates. I digress. What's up, porcupine? Horns? You wouldn't call those horns? I would call them quills, but same thing, different thing, doesn't matter, but what about something else? Horny lizard, what do you call them? The bearded lizards. Call them spines, but still. Just examples of morphology, how things appear. And morphology is really what we think about when we think about phenotype and what proteins are doing, how proteins are functioning. With molecular traits, we are talking about the genotype. What mutation is there at a certain location? How many of a certain thing that we're gonna talk about more as we go along. So we do see differences in size of DNA molecules or specific DNA sequences that will affect the molecular trait that we can look at. And one thing that's the story we're trying to get you to think about is molecular and morphological. Sometimes a single trait can be multiple of these things. And so it all depends on how you look at things. And so a morphological trait could have an underlying molecular trait as well. That's the general idea of what we're trying to get you to look at. And one of the things I mentioned, hopefully on Monday, is that a lot of the sort of the breeding has been done already with looking at if you take plant X and plant Y and put them together, what happens? That's the idea of classical genetics. So Gregor Mendel and all the people that followed in his footsteps just did a bunch of breeding experiments looking at morphological traits. Are they pink flowers? Are they red flowers? And when we typically look at morphological traits, we see a certain type of pattern emerge. And that pattern is usually in the form of either what we call complete dominance or incomplete dominance. And if you don't know what those two things refer to, that's okay. We will hear about them very soon, possibly today, definitely on Friday. But it is important to think about some traits do typically have different inheritance patterns or ways in which they interact with each other. In molecular and morphological, we do typically see some differences. And typically when it comes to things today, we do a lot of DNA sequencing and DNA sequence analysis and comparisons. And when you're looking at DNA sequences, we see a lot of ideas of more codominance. And what we need to understand and hopefully appreciate about humans is we are diploid organisms, like all animals and all plants. Although some plants do have a non-diploid state, but I'm not Dr. Kruger, and this is not the ecology of plants or anything, so you can learn about how plants are different, how they're weird by taking a class with Dr. Kruger or even Dr. Greenwood or Dr. Ray. We have several botanists. As a diploid organism, we have what we call homologous chromosomes. Half of our genetic information came from our father, half of our genetic information came from mother or from one parent or the other parent. So we have two copies of everything. And because they are not really talking to each other in the DNA chromosomes, that's why they're more likely going to exhibit a codominance, because they're both always there, and one is not overriding the other. For the case of complete dominance and incomplete dominance, proteins are being made and proteins are interacting with each other, which is why we do see a difference. The DNA sequences typically will be codominant because they're both always there. But how proteins function together is this idea of complete dominance and incomplete dominance. And the example that was used in the textbook, I should have talked about this on Monday, but I always forget to talk about the textbook. Don't buy it unless you really want to. It might be actually worse to buy it because I've changed the order of things and combined a few chapters together and added things from outside the book. We're required to have a book, and this is where a lot of my stuff comes from, so that's the book I derive a lot of my information from. So if you bought a book and don't really want it, take it back, get all your money, but you get all your money back right away. If you're not gonna read it, oh cool. But I don't assign reading out of the textbook or ask you to do problems from the textbook. Everything you need for quizzes and exams is from the lecture content, those slides and whatever I end up drawing on the board. And so the textbook talks about one of Gregor Mendel's traits that he looked at. And P, shape, was the thing that we're looking at now. Might have to know what to look at, but this is not a tennis ball. This is actually a pea. You need to go to the cafeteria today and have peas. They're pretty good. Pea, fried chicken, mashed potatoes, peas and carrots. Pretty good. One of the things that Gregor Mendel looked at when he was trying to determine the inheritance patterns was he saw that sometimes you have wrinkled seeds, sometimes you have round seeds. And because he was born in a time when DNA had not been identified yet, he couldn't really go very far. But just based on morphology, he was able to say that these two things are different. And based off of the experiments he did, this was dominant and this was recessive. So if you were to combine these two genes together, we would see a certain type of offspring appearance. And we're not going to get into talking about how Gregor Mendel does stuff just yet. Just we have two things, a round that's dominant, and a wrinkled, that is recessive. In this case, it is a story of complete dominance. So the dominant allele is always going to mask the recessive allele. This is a morphological difference, something that we can easily see without going too deep into the organism. It turns out there is also a molecular phenotype associated with this morphological phenotype. And this is something that will impact us as we go forward. Sometimes genes get disrupted. And in this particular case, why the P is wrinkled is an extra bit of DNA inside this particular gene we call a transposon. So in this case, the transposon is this red box. So this is a foreign piece of DNA getting inside of a gene. Remember, genes code for proteins. And if you remember a little bit about biology 140, we go from DNA to RNA, RNA to protein, there's something called a codon and a reading frame. And if you disrupt how the three letters are joined together, you screw things up. So this basically disrupts the gene and makes a non-functional protein. And so in the dominant one, there is no transposon. In the recessive one, there is a transposon. So that basically explains why it's wrinkled. Because the gene that makes it round is now not working. There's a gene that makes the thing round. Actually not, there's underlying biochemical reasons for it being round. But the idea of a piece of DNA getting into a gene and causing a non-functional protein, the result is a pretty important part of genetics. Because we can put something in anywhere and we can disrupt something. Typically, that always is going to result in a non-functional protein. Transposons are natural. We'll talk more about who discovered them and the different types of transposons as we go forward. But the way this was identified was doing a technique called DNA gel electrophoresis. It's a way to separate and visualize DNA based off of size. In molecular lab, you'll definitely run some DNA gels. But the basic idea is if you take this little thing that, if you look at it top, it sort of looks like this. If you look at it from the side, it kind of looks like this. It's sort of this weird kind of goo. It's not gel exactly, but similar type of consistency. And what we can do is we can put DNA into what's called a gel. And when we apply an electric current, the DNA that's in these little what we call wells will move. And the distances these move is based off of their size. I'm not going to talk about exactly how it works, but it is kind of interesting and hopefully useful to understand that bigger things move slowly and smaller things move more quickly. Because we're able to sort of get through the little small holes. It's like trying to get through a hedge. If it's, not a big hole there, it's kind of really hard to kind of squeeze through a hedge, but if it's a really big hole, you can just go through it without any trouble. Now DNA is negatively charged, so it's going to move towards a positive charge when we do gel electrophoresis, because it's an ionic interaction. But this idea of small versus large is a little bit more important idea as we go forward. This because it allows us to, we're going to look at some pictures later on in the semester, and size is kind of important. It's kind of important to know that if you're down here, you're more small, and when you're up here, you're more big. Looking at these things is pretty important. I'm say pretty important. We'll just leave it there. Does anybody like to watch shows like NCIS or CSI or any of those other forensic science type of shows? They will sometimes show things that kind of look like this, where you have these little bright spots on something or a picture. Like, here's DNA. Look, you can compare the DNA of individual A and individual B, and you did it. You're guilty. Your blood sample matches the blood sample of the crime. Now, what is fun about the molecular trait and how we do see a difference between morphological and molecular, there are two types of round seeds. One that has a molecular phenotype like this, and another one has a molecular phenotype like this. Now, if you don't remember anything about 140, maybe you remember the idea of homozygous versus heterozygous. And in Gregor Mendel's experiment, you take two homozygotes, breed them together, you get a heterozygote. And so this is the case that what we say is codominance, because both the big allele and the small allele are both present. Now, this is still it's homozygous, so it's neither going to be any type of way. But the cool thing about it is, here we see that even though the morphological trait, or the phenotype of those two, both round, have a different molecular phenotype. So just because you look the same as somebody else, doesn't mean you're exactly the same as that person, or the thing. So morphology is somewhat useful, but it's not really going to tell you a whole lot about how, or if you are, really different than somebody else. That's why DNA is a really powerful tool when it comes to analyzing things and comparing things. But looks the same morphologically, but also looks different when you look at the molecular phenotype that's present. And just for fun, the study was also done to look at the enzymatic activity of the gene that's involved here. And you don't need to memorize this, because I'm never going to ask you what does SBE1 do. But it makes starch, or actually makes a form of starch called amylopectin. It turns out that if you don't make amylopectin, then you make wrinkled, for whatever reason, because you can't hold water well enough. So if you're round, you have active SBE1, so you're going to be round. If you're wrinkled, you have a transposon in your SBE1, so you're not going to be very good at making amylopectin. And you can see that by measuring the ability to make amylopectin, which is shown here. Now the question you need to think about is we have a homozygous dominant, and a homozygous recessive, and the heterozygous in the middle. When looking at the size of the DNA markers, it was a codominant example. When I was looking at the morphological trait of round versus wrinkled, it's a complete dominance idea. And we haven't talked about incomplete dominance yet. Does anybody know anything about incomplete dominance? Sometimes we don't talk about it in Biology 140, sometimes we do. The idea of incomplete dominance is you get a mixing of A and B together, basically, making something sort of in the middle. Does this look like you're getting something in the middle based on these two graphs, or these two graphs? When both genes are disrupted, you get no activity. When both genes are undisrupted, you get this much activity. When one gene is disrupted and the other isn't, you get this much activity. Does that sound like incomplete dominance, where you're getting a intermediate phenotype? That's the trap, unfortunately. It's actually a molecular trait that's showing codominance, because you're getting the 0 of no activity plus the 80% of activity. No, it's not 80%, it's half of 80%. Because we have one active gene making one active protein, and that's how much activity you get from one protein. When you have 2 copies of that protein, you get twice as much activity. And this is an idea we call gene dosage. It's a pretty important concept. As we go forward, you'll hear much more about it. But just to sort of add a seed for later, gene dosage is an important consideration. You have 2 active copies of a gene, you only have 1 active copy of a gene, or you have 0 active copies of a gene. This is going to be an important consideration. And one place that it really matters is when we talk about males versus females. And just to give you a preview of where we're going to go, if you don't remember the chromosomal idea of male, we attribute it with having an X and a Y chromosome. Female in humans is 2 X chromosomes. So here we see is a problem. Having 2 X chromosomes in females, only 1 inch chromosome in males, means there's going to be a difference in how much X is made. And there's a way to solve that. It's a fun little story that we'll get to. But sometimes we do see these problems with gene dosage, because the chromosome you got from your one parent might inherently make more of a protein than the chromosome you got from your other parent. And that imbalance makes you a little different than the person that's next to you. And it's not always about what DNA sequence you have, it's about how much of something you actually make. And so really you can basically ignore anything about this round versus wrinkled, except for when it comes to understanding the idea of DNA gel electrophoresis and how we use it and where do small things and large things go. As we go into this next set of slides, we are going to dive into complete dominance, incomplete dominance, and codominance, which are examples of simple traits. And then we're going to look at all the different types of, or at least some of the different types of complex traits that are out there. Before moving forward, does anybody have any questions about anything so far? Again, gene doses, don't worry about memorizing it just yet. We'll get to it eventually. But it is going to be important. Well, typically when we talk about the father of genetics, it means Gregor Mendel. But some other papers I've read and other textbooks I've looked at mention somebody else first. So why not throw in another scientist, Carl Reuter. I'm not sure if I pronounced it fast. The reason why I think this is interesting is his results or conclusions are a little bit different than what Gregor Mendel proposed. Now, it might have been the organism he used, but who knows? But Carl Reuter used tobacco plants, and he saw that his offspring had intermediate phenotypes, which is typically what we attribute to incomplete dominance. Now, what Gregor Mendel is going to do and what he's going to publish really is about complete dominance. Now, the other thing that's kind of interesting about what Carl Reuter said is there is a blending of inheritance. So you take what you have from one parent and the other parent, they come together to make you. So however that works, that's you. So you'll never be exactly like mom or exactly like dad, you'll be halfway in the middle. At least that's what a blended inheritance would say, is you get this combination that comes together. Now, we know that's not true, at least it's not true all the time, because there are other patterns of inheritance. What's also interesting is this is basically Civil War era. Right? United States Civil War. You know when Civil War started or ended? 1780 something. Right? Who's taking U.S. history? Come on, you people. Oh well. But if we flash forward 100 years, which is also why it's sort of interesting to think about Paul Reuter, is he was doing genetic experiments well before Gregor Mendel was doing genetic experiments. And I wonder, did Gregor Mendel have any idea of these earlier genetic experiments? I imagine there were papers published that were easily obtainable, but who knows? If you didn't know, Gregor Mendel was a monk, and monks don't have anything else to do, so they count peas. They count a lot of peas. And his experiment took 8 years. Who wants to spend 8 years doing an experiment? I haven't either. It's like a waste. Most of his years was actually done in preparation, not actually doing the experiments. So making sure his parents or starting plants were actually correct. But what Gregor Mendel proposed, based on his analysis of pea plants, is that he proposed the principle of segregation and a second principle we'll call it the principle of independent assortment. We will talk about what these mean, what they say in a little bit, but we'll first talk about what Gregor Mendel actually did and how he did it. That has some bearing on what we look about and how we think about things. One of the most important things about Gregor Mendel's experiments was his parents. He always started or used what he called true breeding, or what are called true breeding individuals. And we define a true breeding individual as if you self-pollinate or breed it to itself, the offspring will always be the same. You never see any variation in offspring. It's something that is only possible in plants. Because plants have both male and female reproductive organs, you can easily self-fertilize, or take two plants and mix them together and it works out fine. Also, you can get a lot of offspring very easily with plants, something that's really hard to do with animals. And plants make really good genetic models, at least starting out. So if you take a true breeding plant with one phenotype and mix it or breed it to a true breeding plant with a different phenotype, what happens? That's the question, what happens? I did two different types of experiments. The first type was called a monohybrid cross, where the parents only differed in one trait. Sorry, one phenotype. Whether it's flower color or seed color or seed shape. There's only one difference between parent A and parent B. This gave him a basic framework for what he thought was going on. But to make sure what was really going on, what he thought was going on, he did what's called dihybrid crosses also, where parents now differed in two traits. So you could take yellow seed, round seed parent with green and rainbow and see what happens, or mix any two things together. And the dihybrid cross is probably a little bit more important than the monohybrid cross, because it does show us some really important things about how DNA is inherited and transmitted to our offspring. But also the monohybrid cross does help us also. And not only did he do these monohybrid crosses and dihybrid crosses, he did a few other things that we'll talk about in a second, like a test cross and a back cross and long things like that. Things that I'll continue to try to tell you and remind you of, things that you can't do with humans, can't easily do with animals. Very easy to do with plants. You can manipulate plants all you want. You get a lot of offspring on plants, things that are really hard to do with humans. Force people to breed and to get a lot of offspring. But what Gregor Mendel was doing is dihybrid and monohybrid crosses. He used terms to describe the type of trait he was looking at. And he called them quantitative and qualitative. The quantitative traits he defined as often difficult to define. Qualitative traits were things that clearly stood out and had definitive differences. Somebody define quantitative in the scientific world. What do you typically think of? Right, something you can actually measure. Something that's difficult to define, is that the same as something you typically measure? Well, one is measuring is define what it is, right? But that's also the point of quantitative. It requires a measurement. And if you're measuring things, at some point you have to say that this is one size, this is another size. Color might be very easy to distinguish, but Gregor Mendel was looking at size of plants sometimes. And something that, not clearly one thing or clearly another thing was a quantitative trait. And he tried to ignore sort of all of the quantitative traits, just look at things that were qualitative. Things that are very easy to say it's A or it's B. There's no in the middle kind of category. And when he was doing his work, mostly he found that the traits followed a complete dominant example. But some of the papers I've read about Gregor Mendel's studies suggest that his data also contains examples of incomplete dominance. He just didn't put it in his main paper because it didn't fit the story that he was trying to talk about. So, but he did say that he did sometimes see things that didn't fit his pattern, but most of the time, this is the pattern that he saw. So And so what we do in science sometimes go with the most common thing and sort of ignore the exceptions. So here we are, quantitative versus qualitative traits. He looked at a number of traits. I'm not going to ask you to memorize all these different types of traits or the different phenotypes that go with these traits. But he did a lot of work. I mean, just doing one of these will give you good information. Just doing 2 of them will probably be enough. But doing 7 different monohybrid crosses, and then doing dihybrid crosses. How many different combinations are there? Mixing 2 together. 2, 2, 2, 2, 2, 2, 2, 2, and 2. And how many different ways to put these together? Lots of different ways. So he was able to do a lot of different examples. And when it comes to genetics, numbers are important. The more time you do something, the better your data is, the more confidence you have in your conclusions. Doing something once could be chance, doing something twice could still be chance, but 10, 20, 100 times, it's hard to see a pattern emerge. So we always start with true breeding parents and we mix these together. So they're always different, at least the monohybrid crosses, we have one difference between our parents. Gregor Mendel did something that's also really important for his analysis. He followed these plants through 2 generations. F1, or first filial, is the first set of offspring, and F2 is the second filial, or second set of offspring. In case you care, filial or filius is Latin for sons. So the first set of sons, the second set of sons. And to get the first set of offspring, you mix the parents together. To get the second set of offspring, you use the first set of offspring as the parents. So you can either self-fertilize one with itself, or use one F1 and another F1, breed those together to get your F2 generation. So what did Gregor Mendel see? With the F1 generation, he always saw the same type of result. All of the F1 offspring had a single phenotype that matched one parent and not the other. So this was not a blending at all. One showed up, the other was completely absent. He did a lot of replicates, so it's not like it was, a fluke, possibly. It happened all the time, regardless of what trait you looked at, always the same. If you take these F1 individuals, and you breed them back to yourself, we're going to get our F2 generation. And that's where we started to see some differences, and that's the thing that really gave Gregor Mendel the information he needed to describe the process that he thought was going on. Again, one of the other things we have to take a step back. DNA had not even been discovered at all or detected in cells yet. DNA didn't exist. Gregor Mendel talked about factors. Parents gave factors. A factor controlled whether you had a purple flower or a white flower. A factor made you a green seed or a yellow seed. And those factors were transmitted to the offspring. You can physically see the trait of one parent show up in the offspring. This idea of this transmission was important. But the problem was, where did the other factor go? Is it gone or is it still there? If it's still there, why doesn't it show up? I mean, know the answer now, but back then, all Gregor Mendel had was math. So, yay, math! Without math, there is no science. At least there is no genetics. Based off of what Gregor Mendel saw with the F1 generation and also the F2 generation, he proposed the idea of dominance versus recessive. And he decided that the dominant trait, or the dominant phenotype, would always be the one that showed up in the F1 offspring. And his idea of recessive came about by looking at the F2 generation. And so when we do all of our monohybrid crosses, not only do we get the dominant trait or phenotype, we also get the recessive phenotype. And so his idea was that the recessive phenotype, the factor giving the recessive phenotype, was still there in the F1 generation, but masked. And it was only allowed to show up later when we did the F2 generation. Make sure you memorize all these numbers. This will be the next quiz. What are all the numbers and what are all the phenotypes that go with those numbers? No? Can you imagine trying to count more than 7,000 P's and distinguishing whether they are round or wrinkled? Can you imagine trying to count more than 8,000 P's and distinguishing whether they are yellow or green? I mean, what's he gonna do? He's a monk.
He must have had a lot of time on his hands.
The question is, did they eat them afterwards? Or did you save them? That's why I didn't hear what you said.
I just said he must have had a lot of time on his hands.
Yeah, he had 8 years to spend. Maybe he was one of those monks that took a vow of silence to let them talk and tell the plants. And so what you also have to keep in mind is the plants that were used for this experiment were different than the plants that were used for this experiment. Because on monohybrid crust, you can't use these or these. So how many different plots of pea seeds did Gregor Mendel have? At least 7 for each of the different traits he was looking at. That's a lot of space. How many peas can you get from a single pea plant? I wonder. How many peas in a pod? 2, right? Because that's the expression. We're like peas in a pod. Or is 3? Is it 3 peas in one? Can you have 3 peas in a pod? Or 4? 4. Anyways, so why is it always much more of one and less of the other? Well, if you do the math, Gregor Mendel did the math, and he found out that was almost always the case, is the dominant phenotype was about 75% of all the offspring, and the recessive phenotype was about 25% of all of the offspring. That's where we get this 3-to-1 ratio that we typically attribute with a complete dominance example. Now, Gregor Mendel didn't use the words homozygous or heterozygous. Those words showed up much later on in literature. They definitely talked about something being recessive versus dominant. Now, he didn't know that the plants were diploid, but he didn't know that the purple flower plant gave the purple factor and the white flower plant gave the white factor. And when you do the F1 generation, only the purple factor is the one that shows up, because it's the dominant one. But the white is still there, it's just not observed. And we know the white is still there because it shows up in the F2, so it's just sort of hiding. And that's why this is called a complete dominance, because you don't see the one, you only see the one. And I use the one twice, but hopefully you pay attention. You see one, but not the other. So with this idea was born the concept of complete dominance. A 3-to-1 ratio in our F2 generation. And that's the important thing. All of the F1 are exactly the same, both morphologically and molecularly. Molecularly. Just to make fun of myself. My first semester here, which was a long time ago, couldn't say the word characteristic in lecture. Added an extra syllable. So also so long I said the word property instead. Second semester I was fine. I could say characteristic all you want, so you didn't say it, right? Characteristic. And they have like a word like that, like always mispronounced. Like skeptical. Skeptical. Fine. Would you like some bad puns instead? We have plenty of those. I believe everything on this slide is something that has already been mentioned, but it does say something new sort of at the bottom. Law or principle of segregation. I don't want to use the word law exactly. I should have changed this because we don't want to confuse thermodynamic laws with the laws of segregation. So it's not really a law, it's more of a principle. But from doing the F1 generation, and if we believe what's going on, that both parents are giving a factor to their offspring. The white factor is in here somewhere, or in all of these, the white factor is present. But when you take your F1 offspring and you make the next set of offspring, you can separate the purple factor and the white factor from each other, which is what allows for the white flowers to actually show up. So we need to call this the principle of segregation. If there are two factors present, those two factors can be separated from each other, leading to offspring with different phenotypes. He didn't really exactly know why it was 3-1. That was sort of explained later on. But here we have a basic idea that factors are inherited. They're not lost or changed. White is still white. It's not changed when it goes through F1 to F2. It's still there. It's just, absent, or not absent. It's just silent, missing, being completely dominated. So it's like it's being sat on the whole time. I will never ask you to write down the actual description of the principle of segregation. It is fairly simple. Factors are separated when we make the cells that are used to make offspring. I know it's kind of convoluted, but you can't use the word DNA and genes. I'm not sure if the word gamete was actually in there. But if there are two factors present, then those two can be separated from each other. And then sort of a little bit later, this idea was sort of changed, written this way, where each organism contains two characters, which we call diploid now. So there's two copies of every gene, and one chromosome is separated from the other chromosome when we go through the process of meiosis. So segregation is that the factor controlling one phenotype can be separated from the factor controlling the other phenotype for a single trait. These next couple slides are just some slides or images from some of Gregor Mendel's actual work. And he was sort of doing some of the math that we talk about later on in the semester, where, does anybody know any German? Well, pollen, I'm guessing, is pollen. And Keim, I think is not pollen. I believe this is male and female, but I guess it really matters. Plants have pollen, and then the other thing that's not pollen is what? Who's taking botany? Who's going to take botany? Anybody? What will you learn? You can tell me. But if you do different types of mixing, and what is trying to be explained here is if you take a true breeding parent and with, like say, purple flowers, you're always going to get purple flowers back, but you're going to have both one factor from one parent, one factor from the other parent. If you mix them together in any way where you take the male purple with the female white and the female white with the male purple, what do you get? And that's a fun little story that we're going to talk about, because he did do sex changes when he did his experiments. Does anybody sort of, what does this look like to you? A plus 2 capital A, lowercase a plus A. If it were x squared plus 2xy plus y squared, it's a, well, it's sort of the same idea as the binomial equation. He wasn't quite there yet. He didn't call it 2 capital A's. But that's the way we think about it now. We think about it as a binomial equation. X squared, y squared, you get two X's and two Y's, or one that's XY and one that's YX. Now, there's another figure from this paper, which I think is also pretty interesting, is he went crazy with putting things together in different ways. And again, this is something you only do with plants. There are laws that prevent some of these things from actually occurring. Parents aren't supposed to have sex with their children. I say aren't supposed to, right? It's a law, right? Incest is against the law. Some people sort of, maybe it's not supposed to joke about incest. Maybe that's it. Maybe that's the problem. I'm the asshole here. It's probably true. But back crosses are an important part of what Gregor Mendel did to take his F1 generation, breed it back to the parental generation, and see what happens. Or take the F2 and breed it back to the parental, and see what you get. That's sort of what's in all this, is that idea of taking F1 and a parent, breed them together, take F2 and parent, breed them together, and take the other parent, breed it to the F1, and the other parent, bring it to the F2, see what happens. Because you have all different possibilities of mixing and matching things together. And today we call true breeding homozygous. And we use homozygous to mean that you have two copies of the same allele, which is why you are homozygous. And the parents were all homozygous, but the F1 generation is now we will refer to as being heterozygous, which is meaning you have different copies of a gene, so you have two different alleles present. So this explains why the white can still be there, the white factor is still there, but the purple is the only thing that's actually going to show up in our offspring. So homozygous, heterozygous are important terms when thinking about the experiments they did. Basically, all of the F1 generation is a heterozygous or are heterozygous individuals. Both parents are homozygous individuals. For the F2 generation, we can talk a little more about what those actually look like, because there are some differences. But I've talked around it for a while, but some of the other things that Gregor Mendel did were these back crosses, which is taking parent to offspring. He also did reciprocal crosses, which is important, and making sure that it's not a sex-dependent trait. So if you switch the sex in the parents, so an example would be here, just red male, blue female, and then switch it, make, sorry, blue male, not blue make, and red female. He also did some test crosses, which is a way that we can identify a genotype of an unknown individual. But when you do a test cross, one thing you need to make sure you do is that you always use your homozygous recessive parent as the other partner in the breeding experiment. So doing back crosses and test cross and reciprocal crosses, you can see does the pattern hold up, and you see different patterns. When you take the F1 and breed it to the parent, you're going to see different types of things in your offspring. And we'll look more about Punnett squares and how we deal with Punnett squares as we go forward. Now, if you're really ready for a Punnett square, here it is. You have to wait until Friday to talk more about what this means and how we set it up and what we can do with it. We're not all about Punnett squares just yet, but that's help us think about Gregor Mendel's experiments.